everyone. I'm Jake Vogel from Lund University. I'm so sorry I can't be with you today. I've had some rather unfortunate difficulties traveling to Seoul. With any luck, while you're watching this video, I'll be in the air en route to you. But I'm sorry I can't be here to deliver the talk in person. But a huge thank you to Kaio for putting together this really interesting interdisciplinary session. And thanks to the organizers for selecting it for an OHBM symposium. So my role here is to talk about connectome-based models of neurodegenerative diseases, mainly Alzheimer's disease. In keeping with the theme of the symposium, I want to talk about work not just studying how the connectome changes in these diseases, but instead to actually leverage the connectome toward learning new things about the diseases and perhaps even building clinically relevant tools or resources. So my lab is very interested in Alzheimer's disease, and it's important to understand it has a very complicated pathophysiology. So Alzheimer's is characterized by the aggregation of two different pathological proteins in the brain, amyloid and tau. Amyloid builds up through the cerebral cortex many years before any cognitive symptoms manifest, as you can see here, becoming quite saturated across the cortex fairly early on in the disease process. Now, in contrast, tau builds up quite focally in the entorhinal cortex before spreading in a very particular sequence through select regions as the disease progresses. Now, fortunately, we can measure both of these pathologies in living people using positron emission tomography, or PET imaging. Now, particularly in the case of tau, many researchers, including myself, have been quite fascinated about this very particular pattern of aggregation. What causes this specific pattern? Why does it happen? And what can we learn about the disease from it? So one hypothesis actually emerged back in the 80s from neuropathologists looking at histochemical stains of postmortem brain tissue. So just from observing these slides, the pathologist concluded, and I quote, "These data support the suggestion that the pathological changes in Alzheimer's disease affect regions that are interconnected by well-defined groups of connections, and that, emphasis mine, the disease process may extend along the connecting fibers." So this was really the origin of the network spread hypothesis of neurodegenerative disease, which suggests that pathology can actually use the brain's intrinsic network architecture to spread, almost like an infectious disease uses a highway or airways to spread through the world. So fast forward 15 years, and this hypothesis really started taking off thanks to a seminal study by Seeley and colleagues. So shown here in these blue blobs superimposed on the brains are brain atrophy patterns of 5 different forms of dementia, each with very distinct cognitive and behavioral symptoms. So Seeley and colleagues identified the region of peak atrophy for each of these maps, which are shown in little white circles, and used this as a seed for resting state functional connectivity in young healthy people. And they found that the resulting functional connectivity maps look qualitatively very similar to the atrophy maps in a manner that was actually rather specific to each form of dementia. So this raised the idea that neuropathologies were targeting specific intrinsic behavioral brain networks. So this paper was a huge proponent in sparking this network degeneration hypothesis. And that's a hypothesis that I'm proud to say has been pushed quite substantially by the neuroimaging community. But if you want to think about this mechanistically, how does the network-based spread of disease actually occur? So one hypothesis is that neuropathologies like tau and Alzheimer's actually physically spread from neuron to neuron, making their way down axons and spreading across the synaptic cleft to the postsynaptic neuron. So if that's the case, we should be able to see this pattern represent even at the macro scale that we can image with neuroimaging. So if we assume pathology starts in one place, in an epicenter, that means that regions that are more connected to that epicenter should show more pathology or early pathology compared to regions that are less connected to the epicenter. So this is a hypothesis that we can actually test in humans with neuroimaging data. So how do we use that? So we do this using connectome-based models. These models represent the brain as a graph, as you can see here, where individual nodes represent brain regions. And in those regions, we can use PET or MRI to represent regional neuropathology or atrophy. So the edges of this graph can be represented by brain connectivity, which we can measure with fMRI or diffusion-weighted tractography, which I think you're all probably quite familiar with. So using this framework, we can build models or run simulations that test the degree to which brain networks influence the distribution of neuropathology. So I want to focus on one particular species of connectome-based models, which are called connectome-based diffusion models. And in this model, which is actually more of a simulation, we establish an epicenter region and we inject a signal into this region. We can then simulate the diffusion of this signal through the brain's intrinsic network over simulated time, and we can measure how well this simulated pattern represents the true pattern of pathology distribution in the brain that we measured with PET or MRI or what have you. So we ran a version of this model called the epidemic spreading model, which was actually introduced by Yasser Medina, and we used that to predict the spread of tau. And here's how that model performed. So here, each dot is a brain region. The x-axis represents model simulated pathology, and the y-axis represents the true pathology. So you can see pretty clearly across the population, this model did a remarkable job predicting the distribution of tau in the brain, which really supports the notion that tau is using axonal connections to spread through the brain. We also measure the performance of this model using various different brain regions as epicenters, just to check. And if we look at the international staging system for tau pathology, or the Brock stages, we can see that the very first stage involves tau accumulating in the entorhinal cortex. Sure enough, our model achieved best performance when using the entorhinal cortex as the model epicenter. So this really lends credence to the model accurately recapitulating this disease spread process. Now, of course, we were not the only ones to find this. This finding that tau patterns are influenced by regional connectivity has now been replicated by at least 5 different labs, probably more, using slightly different approaches and even using different data sets. So altogether, what do these studies tell us? Well, I think what's clear and what we can summarize from these points is that in Alzheimer's disease, tau appears to be distributed in a pattern that resembles brain networks. And really, these studies strongly support the network spread hypothesis, especially with respect to tau in Alzheimer's disease. So I think it's important to note that this body of work has actually been quite influential in Alzheimer's research. So in parallel to the neuroimaging studies I've described, basic scientists have also done a lot of work to show that the spread of tau in mammals is mechanistically feasible. So tau can definitely be secreted from neurons in various forms. It can be taken up by postsynaptic neurons. It can be transported down axons. It's not proven that this is what happens in humans in Alzheimer's disease, but it is proven to be physically possible. So this has actually led to the development of pharmaceutical interventions as well. So there are now several anti-tau immunotherapies that are in development. And it's really important to note here that tau inclusions that we've been talking about this whole time occur intracellularly, so within the cell soma. And these immunotherapies, they cannot enter the cell soma. So the whole idea of them working is predicated on this network spread hypothesis, on the idea that tau is spreading across synapses, and the hope is that these therapies can intercept them and actually halt the spread of tau. There's also, of course, plenty of basic research that's still trying to better understand tau spread mechanisms. But I think overall, it's quite exciting that these neuroimaging studies have substantially contributed to this line of thinking that's now driving towards therapies. All right, so what can we get out of these connectome-based models? They were originally designed to test the tau spread hypothesis. I think they've done quite a good job at doing that. But I also think that we can get a lot more out of them. After all, they represent dynamic simulations of the neurodegenerative disease process. And there are definitely ways that we can improve them. One of the main goals, I think, is to glean further neurobiological insight out of these models. So testing more complicated hypotheses, even potentially generating new hypotheses, and trying to understand mechanisms a little bit better as far as we can do with sort of our neuroimaging perspective. It's also important that we try to make these models not just informative, but practical. So can we use them to make predictions about the spread of tau over time at the individual level? Can we do so in a manner that could be useful in clinical trials or other clinical scenarios? So we recently published a review outlining ways that the connectome-based models can be innovated on in order to achieve these objectives. I refer you to the review for details. You can see the QR code, which links to the paper. But for the rest of the talk, I'd like to discuss some newer findings building on these themes. And I'd like to start with some studies that are really designed to generate new neurobiological insight using connectome-based models. To do this, we've recently been using the susceptible infected recovered model, or the SIR model, developed in the labs of Alan Dagar and Bratislav Misic at McGill. So just like the epidemic spreading model, this model also simulates the spread of pathology over the brain's connectome. In addition, it models intracellular mechanisms, namely, as you can see here, synthesis, clearance, misfolding, and spread of pathological proteins. But we do not have to assume that these parameters are the same in every brain region. For example, why should we assume that the synthesis rate is the same between two brain regions that we know express different levels of the protein of interest? We shouldn't, and we can actually add a priori regional information like this to this SIR model, allowing it to moderate individual parameters on top of the connectome-based spread. And you can see that those individual parameters listed here in red: spread rate, synthesis rate, clearance rate, and misfold rate, all of which can be influenced by a priori biological factors that we can basically put into the model. So this was initially used to model the spread of alpha-synuclein, which is the protein composing the main pathology of Parkinson's disease. The model showed that the brain's network architecture was indeed informative in predicting the spread of atrophy, this time in Parkinson's disease. But the authors next asked if the regional expression of key Parkinson's risk genes might improve the ability of the model to predict atrophy in Parkinson's. So regional expression of these genes was provided by the Allen Human Brain Atlas, and including this information as an a priori moderator of local regional clearance or local regional synthesis of alpha-synuclein, it actually substantially improved the model fit, which you can see here in these red lines. Now, it also gives us some insight into how these genes mechanistically interact with alpha-synuclein, where in this case, GBA might be resulting or used in clearance, and SNCA might be more involved in synthesis. So recently, a postdoc in my lab, Xiao Yu, applied the same framework to predict the spread of tau pathology in Alzheimer's disease. So shown here is the regional observed distribution of tau pathology. Fitting the SIR model I just showed you to this data, showed the model very quickly resolves at an accurate prediction of tau pathology, especially early tau pathology. And that's really just using brain connectivity. So this is really a replication of the study I showed you earlier. But of course, this model is not perfect. It underestimates tau in certain brain regions. It overestimates it in others. So we know that tau is encoded by the MAPT gene. So using the SIR framework and the Allen Human Brain Atlas, we decided to use regional MAPT expression as an a priori moderator of regional tau synthesis. And doing so resulted in an even better model performance, and it particularly improved prediction of later stage regions. Interestingly, we saw a pattern where the model once again quickly reaches a high performance level, but now continues to improve over time. So in conjunction with other recent studies and other results that we have, we think that this indicates that low-level tau is actually spreading quickly through the brain, but then intrinsic regional factors influence local production thereafter. And the latter is probably variable across people, but I won't get into that for now. So we use this framework to test a few known hypotheses of Alzheimer's disease mechanisms, but this time, in using human data. I already showed you how MAPT synthesis just generally improved prediction of tau on top of intrinsic functional connectivity. We also showed that allowing regional amyloid pattern to influence spread also improved the model, but specifically in people with biomarker evidence for brain amyloid. And finally, the ApoE4 allele is the leading genetic risk factor for Alzheimer's disease. We showed that allowing regional ApoE expression, gene expression, to moderate tau clearance improved the model, but specifically for people carrying the harmful ApoE4 allele. But we can go beyond just testing known hypotheses, right? So we can also generate new ones. Over the last few years, the Misich lab at McGill has curated and released several really remarkable data sets that represent biological properties of the brain. So we now have spatially resolved information for diverse features ranging from metabolism to specific receptors in the brain, in addition to different ways of representing relationships between regions, not just connectivity, but intrinsic biological similarity between regions. So Xiao Yu, the postdoc in my lab, decided to run a whole bunch of exploratory simulations testing whether certain regional features, such as cell types or neurotransmitters, other cortical features, might help to predict the topography of tau accumulation in Alzheimer's disease. While we have thus far assumed that tau spreads through anatomical connections, and there's a lot of reason to assume that, Xiao Yu also decided to test other ways of representing relationships between brain regions. So this busy plot here summarizes the results of all of these models. I could obviously spend the entire presentation going through all of these interesting results, but I want to draw your focus to one finding in particular. So let's forget about the biological features for a second and focus on the different connectivity measures we have here. So here are the models using structural connectivity, and this line represents the performance of a simple structural connectivity model without any biological factors. This line here represents the performance of the best structural connectivity model, so one that also uses tau information to influence the model. Interestingly, the structural connectivity models perform better than all of the other models using other forms of connectivity, except one, which is regional neuroreceptor similarity. So in other words, the profile of receptors shared by two regions is a better predictor of tau accumulation than how physically connected those regions are. So this is a rather contentious finding. It's got a lot of different possible explanations, but we're working hard on continuing to explore this. All right, so I want to wrap up the talk with just one more example of how connectome-based models can be used to advance our understanding of neurodegenerative diseases. So I think it would be great if we can make these models practical and use them to forecast future tau accumulation at the individual level. We've been working quite a lot with Pavan Chagar and Alain Gorielli at Oxford. And Pavan has adopted another form of connectome-based simulation models that's focused a lot actually on making these models more identifiable. The model, once again, does an admirable job, as you can see here, recreating the canonical pattern of tau accumulation in Alzheimer's disease. Now, here we can see each dot represents a brain region, and the left plot is baseline tau levels, the right plot is change over time, and the plots altogether really show how well the model is predicting these two components. And as you can see, the model does an excellent job predicting not only cross-sectional tau, but also regional change in tau levels. That's a very challenging task to accomplish. Not many studies so far have been able to do it. But the real question is, how does this handle out-of-sample data? And that's something that really has not been shown in the literature. So in this plot, each row is a subject, and each column is a brain region, and the last column is the average across brain regions. The x-axis is time in years, the y-axis is tau levels, and each dot is an observation or a time point where we've measured tau PET. So by training on the first 3 time points in blue, we can make a prediction over time, and that's represented by the gray line. So you can see that in most cases, the 4th out-of-sample time point, which is shown in red, falls well within the confidence bands of these model predictions. So we're actually achieving some pretty good out-of-sample prediction, and this is extraordinarily hard to accomplish. Let me tell you. For now, it's obviously a proof of concept and not so practical, because we're not going to measure 4 years of tau data just so we can measure what's going to happen in the 6th year. But we're working really hard on trying to use less data to train these models, and at least this shows that it can be done. Now, the whole time we've really been focusing on just tau pathology, but as I pointed out in the beginning of the talk, Alzheimer's disease is also involving beta-amyloid and neurodegeneration as key components of the disease. So Pavan has now adjusted his model to accommodate all 3 of these components. In this particular model, amyloid accumulates, and along with connectivity, it actually regionally influences where and how much tau accumulates at a regional level. Tau then influences where and when atrophy accumulates. So if you put all these things together and you run this simulation, we can see that amyloid accumulates early to the point of saturation, thereafter leading to accumulation of tau in the canonical regions, finally following regional neurodegeneration in key vulnerable regions. So you can see with just a few small components, the model does a remarkable job simulating the whole disease trajectory. And not only that, but you can see with this bottom row of plots in particular, which represents longitudinal change, it does quite a good job predicting regional change in all 3 pathological components: amyloid on the left, tau in the middle, and brain atrophy on the right. So we're working to further develop this model for individual forecasting, but we're already really quite excited with what we have so far. All right, to wrap everything up, I've hopefully convinced you that the human brain connectivity can at least partially explain the pattern by which tau distributes in the brain, and especially as it pertains to regions that are affected early in the disease process. Now, in addition, connectivity seems to interact with intrinsic and dynamic regional factors in dictating the accumulation patterns of tau. So while I think transsynaptic spread is still the leading hypothesis, and we've shown a lot of support for that idea, we should still continue to investigate alternative explanations. And finally, connectome-based models have the potential to go beyond just hypothesis testing. We've shown a proof of concept that such models can forecast longitudinal tau accumulation at the individual level. And so we think that this can be developed into technology that can aid in clinical trials in the future. So since I'm not here, I unfortunately can't take your questions, but do please feel free to email me or contact me on Twitter, or come find me later in the conference if I ever actually make it, which we don't know at this point. But I also want to note that our lab is looking for a research technician with neuroimaging experience. And we're, of course, always looking for postdocs. So please do contact me if this is generally of interest to you, if you're into Alzheimer's disease and neuroimaging and multi-omics, etc. Most importantly, I want to thank my wonderful lab, you can see here, the Demon Lab, and all of our amazing collaborators and our funders for supporting the work. And finally, I should note that Jonathan Ritmo, who is a PhD student in the lab, will be giving an exciting talk about functional reorganization in aging and Alzheimer's disease on Wednesday, so don't miss it. Okay, well, thanks so much for your attention, and have a great rest of the session, and have a great conference.